Hello everyone, I'm Hyun-Seob, Associate Professor at University of Nebraska-Lincoln. I'll be talking about the development of deep neural networks to infer interspecies interactions from spatial temporal image data of microbial assemblies. This work was done in collaboration with the Soil Microbiome SFA team at Pacific Northwest National Laboratory. Microorganisms in natural habitats, such as soil and sediments, they self-organize into specific spatial patterns as driven by interspecies interactions, which are in turn affected by metabolic capabilities of member species and environmental variables. Therefore, spatial patterns, particularly at microscales, can be viewed as a key ecological phenotype, but their use for inferring interspecies interactions is currently lacking. Conventional network inference methods that rely on the analysis of population density data have not been extended to handle microspatial data yet. So we propose machine learning, specifically deep learning models, as a new computational tool to predict microbial interactions from spatial image data. In the next couple slides, I'll briefly introduce artificial neural net models and deep learning approaches. An artificial neural network model has a typical structure composed of input, hidden, and output layers. The figure in this slide illustrates a shallow network with a single hidden layer. It relates input variables x and output variables y through the three key mathematical operations and components. First, linear mapping, which is a weighted linear combination of input variables. In many cases, this linear mapping alone cannot effectively represent output variables. Therefore, it has biased terms additionally. Simply, they are constant terms. By having biased terms, the system is not linear anymore. It's called an affine system. Finally, it has an activation function, which makes the input-output relationship highly nonlinear. These three components have parameters to determine. As we'll see later, training and validation is a process of determining these parameters through data model comparison. The network becomes deeper as the number of hidden layers increases. When we hear about deep neural networks, most of them are convolutional neural networks or CNNs, which are particularly effective in recognizing image data. The model structure was inspired by hierarchical structure of biological cells, such as the human cortex. Neural nets progressively extract information from images, from simple to complex ones, through multi-layers. In this example of a car image recognition, bottom left, the deep neural network extracts lines and edges in the first hidden layers, parts in the middle layers, and finally objects in the output layers. Various different network architectures have been developed so far, for example, through competition, and are commonly being used, which include the LeNet5, AlexNet, GoogleNet, and ResNet. In this work, we use the ResNet. Modern imaging techniques enable generating high resolution and high throughput images of spatial organization of microorganisms at the microscale. While these images can serve as valuable sources for predicting interspecies interactions, their direct use for training deep neural networks is practically not possible because of the following two reasons. First, we need the labeled data for training the model, but experimental image data are not labeled. In other words, we do not know about the input-output relationships, and therefore they cannot be used for training supervised machine learning models. Second, deep neural networks require sufficiently large data set to appropriately determine model parameters. However, the size of image data obtained from experiments is generally scant for deep neural networks. In this work, therefore, we used high fidelity backcasting model to fill this gap. Individual-based or agent-based models were used as a tool for generating in silico images on the diverse growth and interaction context. For these in silico images, we know, of course, the input-output relationships and the data size is not limited, therefore very much appropriate for training the deep neural networks. The figure below shows how our approach works. We design in silico experiments to generate spatio-temporal patterns of microbial assemblies under predefined interaction scenarios. This labeled data is used to train the deep neural network models, which is subsequently used to infer microbial interactions from new images that are not part of the training data set. This animation demonstrates how agent-based models can generate self-organized spatio-temporal patterns of microbial assemblies as determined by interspecies interactions. Even though initial spatial distributions of two organisms, shown in red and green, are identical, completely different patterns evolve depending on whether they cooperate or compete with each other. When they cooperate on the left, they like each other and they form initial seed clusters and keep growing around those clusters. In contrast, in the case of competition on the right, they hate each other, they try to be far away from each other as much as possible and grow randomly here and there. This result indeed shows spatial-temporal patterns of microbial assemblies are an ecological phenotype determined by interactions, and agent-based model properly capture this aspect. We allocated data set for training, validation, and testing as follows. We generated 5,000 in silico images under homogeneous environments where interaction coefficients are assumed not to change in space, and split them into 80% and 20% for training, validation, and testing. The trained model was then used to predict interactions for the following three cases. First, new in silico images in homogeneous environments that are not part of the training data set. Second, in silico images in heterogeneous environments where interaction coefficients are not constant anymore but change in space. Third, we also tested actual experimentally derived images and then how the deep learning model trained from in silico data works against those. We obtained a highly robust prediction of deep neural networks tested against new in silico images in homogeneous environments. The figure in this slide shows the performance of 15 resonant models we tested. We also compared the result for the best performing network as well as ensemble prediction using top 3 models as represented by green and red dots. Interestingly, ensemble models always outperform the best individual model. AIChE score was very high for 70 to 30% of cell densities, while it dropped a bit after that. However, the decrease of prediction accuracy for low cell densities is understandable because those images do not have enough information content to be used for inferring interspecies interactions. Next, we increased the size of in silico images from 100 micron to 450 micron scales. For these large size images, we could not assume that interaction coefficients are constant because the environmental conditions will be heterogeneous. To handle these heterogeneous cases, we defined a small moving window to scan the entire image through sliding. We extracted a set of constant interaction coefficients from a given window because the window size is relatively small. But through moving, each location is assigned with multiple different values of interaction coefficients. Therefore, as shown in the right-hand side, interaction coefficients have gradients in space. Our prediction was very similar to targeted values in this case as well. This much high performance was surprising to us because we just used a model trained from homogeneous data without further training to predict the heterogeneous cases. Finally, we tested our deep neural network models against experimentally generated images. We designed co-cultural growth experiments with Pseudomonas fluorescens and E. coli mutant in the presence of 2 different carbon sources, A, with chitin polymers, and B, chitin monomer N-acetylglucosamine. The N-acetylglucosamine can be a major substrate that two species can directly utilize, while chitin polymers or chitin beads need to be degraded by Pseudomonas fluorescens into chitin monomers for uptake. Therefore, these two organisms develop the degrader-chiral relationship for the first case, where chitin is the only carbon source, and competitive relationship for the second case, where these monomers are available. Here is the result for the case where N-acetylglucosamine is used as a carbon source. Therefore, two organisms are expected to develop a competitive relationship. In the image on the left, black is the background, and green and red denote Pseudomonas fluorescens and E. coli respectively. Although to the human eyes, it is impossible to tell anything about their relationship from this image, the deep neural network model clearly predicted their interactions as expected. It is shown on the right, where both interaction coefficients AGR and ARG, meaning the influence of R on G or influence of green or red, are shown blue in color, which means their negative influences on each other. Now, I'm showing how interactions between Pseudomonas fluorescens and E. coli can turn into degrader-chiral relationship in a different context, where chitin polymers are used as the carbon source. Pseudomonas fluorescens positively affect E. coli growth, since the heat map in the right bottom panel shows a reddish color, which means the strong positive influence of the Pseudomonas fluorescens on E. coli. In contrast, the right top panel shows E. coli has no significant effect on Pseudomonas fluorescens growth, as indicated by low magnitude of interaction coefficients. But it was interesting to see that influence of E. coli on Pseudomonas fluorescens to qualitatively vary in space, positive in the upper area, negative in the bottom area. This work has recently been published in Computational and Structural Biotechnology Journal by invitation. I encourage the audience to look at this paper for more details. In summary, this work presented deep learning as a powerful tool for predicting microbial interaction from spatial image data. Two major challenges we had to overcome include unavailability of labeled data required for supervision and insufficient data size for deep network modeling. So we used agent-based model as a data generator. Even though we trained deep learning models only using in silico homogeneous data, the resulting models provided accurate predictions when tested against heterogeneous data, including in silico and experimentally generated images. Our work, therefore, provides a generalizable pipeline for developing deep learning models for improving our understanding in many key science areas, including microbial ecology and soil and environmental sciences. This research was supported by the Department of Energy Office of Biological and Environmental Research, and is a contribution of the scientific focus area phenotypic response of the soil microbiome to environmental perturbations at PNNL. Thank you for your attention. If you have any questions on this work, please feel free to reach out to me at the email shown here.